Assalamu alaikum everyone. My name is Haider Khan and the paper that I am reviewing for my third KPR of this semester is nanoparticles with Raman spectroscopic fingerprints for DNA and RNA detection. Really, the technique that the paper describes is pretty much summed up in the title itself. So let's go forward. Okay, so first of all, let me provide you an overview of the technique that the paper describes for detection of DNA and RNA. First of all, the paper describes a technique which involves the use of a three-component sandwich assay, which involves whole nanoparticles, oligonucleotide sequences, and Raman tags. And then you get a silver layer that is formed around this assay, and which is used for the search signal enhancement. And finally, the detection limit that this paper describes for the technique of detection of DNA and RNA is 20 femtometer. Now, just realize that a femto is 1 in 100 million billion. So the detection limit for this paper is the technique described in this paper for detection of DNA and RNA is actually really small. Now let's move forward and I'll describe each of these things in detail. First of all, what we have are gold nanoparticles. Now, even in my last KPR, I described how gold nanoparticles are actually really safe for human body, for any animal's body. They are extremely non-toxic and they are easily biodistributable. So they are being used in a large number for biosensing in the field of medicine, in the field of uh, pharmaceutics, and in any field for biosensing really. And then we have an oligonucleotide sequence, and the way we select this oligonucleotide sequence is that we look for the target DNA first. We see what the target DNA is, and we find an oligonucleotide that is complementary to that target DNA. Now you know that DNA is a double helix, and once you get one strand and it has a complementary strand on the other side, and the oligonucleotide sequence that we choose has to be complementary to the DNA that we are going to detect. Then you have Raman tags. These tags are the particles that emit Raman signals, which can be viewed outside for detection. And finally, you get this assay, which includes the cool nanoparticles, the Raman tags, and the oligonucleotide sequences. Now, I had mentioned that we use silver enhancement, and what this does is that you get the gold nanoparticles, which promote the formation of a silver layer around the assay, which serves to enhance the search signals. So I'll describe it further as well, that without the silver enhancement, the search signals that are emitted are really small in number, so they and really less in intensity, that they serve no practical use really. And so we use silver to enhance the the intensity of these signals so that they can be used for detection. And one thing that should be noted here is that the gold nanoparticles themselves can also serve for the signal enhancement. However, the constraints that we have here is that we are detecting DNA in very minute amounts, and as a result, the gold nanoparticles cannot gather in such a large number so as to enhance the search signals. So we cannot use gold nanoparticles themselves for signal enhancement, and as a result, the gold nanoparticles promote the formation of silver layer, which serve for signal enhancement. So one of the two experiments that have been provided in the paper itself is here. This is where we use CY3 as a Raman tag for DNA detection. So if you can follow my cursor here, here we have the DNA target, which is in a way complementary to actually the actual DNA target, and we use this to attach to our nanoparticle and Raman tag assay. And once the DNA target is identified, This whole assay binds to this DNA target due to the complementary nature of our oligonucleotide sequence. And once the silver treatment is done, then you fire laser, and as a result, you get enhanced search signals. Uh, so here we have the spectrum that is obtained after this whole result, and let me go through the whole sequence, the whole diagram in sequence. Here in the section A, it has been shown that before the silver treatment, you really do not get any surge signals to obtain an image of the detection. So as a result, you first pass this whole system to silver treatment, and afterward you can easily see the Raman tags here. So silver treatment does really enhance this search signals so that they can be seen in our spectrum. And finally, you get this spectrum for a particular oligonucleotide sequence. You can easily see how sharp these peaks are. So you can very precisely detect what DNA is present and where it exactly is. So the reason why this is being called the Raman fingerprint is because you can use this particular sequence uh, that for any experiment that in which you are going to identify a particular oligonucleotide sequence. So let me reiterate that this sequence that you have obtained using CY3 as a Raman tag is consistent with all the experiments that you will perform in order to detect a particular DNA target. So if you are, let's say, targeting a DNA sequence A, then whatever Raman tags you use, these Raman tags can still help you identify the particular sequence A, and you can easily cross match the whole target using this characteristic Raman fingerprint. So this is the role that the Raman fingerprint play for a particular oligonucleotide sequence. Now moving on to the second experiment that has been defined in the paper itself. This includes the virus detection, virus and bacterium detection, to be more precise. Here we detect 5 different kinds of viruses, which include hepatitis A, hepatitis B, HIV, Ebola virus, and Viola virus, as well as another form of bacteria that is being labeled here with BA. And for the detection of these different viruses and bacterium, we use 6 different Raman dyes, each of which have different colors. So in the initial experiment, you get a grayscale picture of all these viruses. First of all, in the experiment 1, you can see that all 6 different material, 6 different organisms, let's call them organisms for now. 6 different organisms were present, although viruses are not really organisms, but let's for the sake of convenience call them organisms. Then in the remaining 7 experiments, we systematically remove these organisms one by one, and you can easily see that the results are pretty consistent. So but however, the disadvantage of this grayscale map is that you cannot see if there has been any cross hybridization or not. And for that purpose, you transform this into the visible region where you get different kinds of different colors. And through these colors, you can easily see that there has been no overlapping. So the no overlapping really means that there has not been any cross hybridization, and that is another success of this paper, that it can not only detect different kinds of DNA, but it can also differentiate between them, also indicate that there has been no cross hybridization. So moving on, I need to highlight one more point, that RNA can be detected in a similar manner. And this paper also shows a detection of 2 different types of RNA using different experiments. So in conclusion, the paper describes a very efficient, a very safe and non-toxic, and a very selective and specific way for the detection of different kinds of DNA. And not only can it detect the DNA, but it can also distinguish between them, and like in the last experiment I showed you, it can also tell if there has been any hybridization or not. So the paper has been cited over 35,000 times, and was certainly a revolution in the field of detection of DNA using nanoparticles. Do note that earlier different fluorescent techniques were being used for detection of nanoparticles. However, the spectra that you used to obtain was so overlapped that it was difficult to distinguish between two different kinds of DNA. However, this paper really describes really sharp peaks of Raman spectra to help you precisely predict where a particular DNA is.